Okay, so now this is the heart and soul of immunity. These terms didn't make the syllabus anywhere, but here's the graph that shows this. If you digest this graph for a little while, they tell you that you should be aware that some lymphocytes act as memory cells and can quickly reproduce to form a whole bunch of identical antibody-producing plasmid cells if that bad guy with a specific antigen is re-encountered. And you have to imagine that it is. When you bump into a particular bad guy, a flu virus for instance, it's not likely that that first time is the only time in your life that you're going to bump into that particular bad guy. But the second time you bump into them, second exposure, you are much more ready for it because of the way your system works than the first time. Okay, so here we go with this graph. Here we have a bad guy that has antigen A. We'll call it bad guy A, pathogen A. You bumped into it on day one, right? If that's 0 and that's 7, that would be like day 1. It looks like it took you 5 days, literally by this graph. That's not every time, but literally by this graph, it took you 5 days to start making antibodies, because here it first shows up the antibody concentration is even there and it's increasing. This specific antibody to this bad guy A. Okay, what's making them? White blood cells called B cells. What are they also doing? Mitosis. They're making more and more of themselves, which is why they're making more and more antibodies. More and more antibodies clinging to more and more bad guys make it more likely that those bad guys are going to be gobbled up by phagocytes. And then at some point in a signal that I don't know if it's still understood yet, your body gets some feedback, some negative feedback saying, okay, you've done enough of this. The bad guy is defeated or mostly defeated. It's time to stop making more antibodies and stop doing that mitosis. So what happens then, you see, is antibodies decrease. Well, why would they decrease? If these antibodies are made, why would they decrease? Because they're sticking to bad guys and being eaten up by phagocytes. That's why they decrease. But what doesn't decrease is those cells that were making them and the cells that weren't actively making them but had the right receptor and the right recipe to make them, those called memory cells. They persist. And that's what, again, the heart and soul of immunity is, the persistence of memory cells. So I remember reading in this COVID thing is just, it's unbelievable, the hysteria that people will buy into. The reports that, well, so-and-so tested positive for antibodies, but then 2 weeks later they had a whole lot less antibodies. So it looks like maybe you won't become immune to COVID. Well, every kind of immune response involves an initial increase in number of antibodies and then a decrease for the reason that I told you. Anyway, so look what happens the second time you bump into bad guy A. You now have all these memory cells, and they go to work right away in a volume that far exceeds what the initial group was able to do because there were so few of them, and it makes so many more antibodies so fast, right? This is time, so fast that the bad guy never gets the upper hand. You don't even know this battle's going on unless you were tested, and that is exactly what immunity is. It's a much faster response to a bad guy compared to the first time you got it. So they throw in here the specificity idea. If you happen to, and of course this is just for graph purposes and explanation, if you bumped into a different bad guy for the first time on that same day that you bumped into bad guy A for the second time, your fight against bad guy A doesn't help you against bad guy B because it's a specific shape. It needs to bump into those specific receptors on specific B and T, and you got to start that fight all over. You're going to make different shaped antibodies to B. So this is the nature of your immune system being specific. And again, since there is a whole host of viruses that cause what we call the cold, including some coronaviruses cause what we call a cold. A cold is just a mild thing that has all the symptoms of sneezy whatever, but over 200 different viruses can cause those same symptoms. That's why you don't build up immunity to the common cold. You build up immunity to specific cold viruses, but since there's 200 of them plus, you're going to get what you call a cold all your life, and it'll seem like the same disease to you, but it's actually a different disease every time. Okay, so now we have vaccines, right? Just a little bit of vaccine stuff here. I'll go over much more in class, but once we learned how all this works, or even way before we learned how all this works, way before we learned about, B cells and T cells, we came to understand that if you get a disease, you usually don't get it again. And this started the wheels turning in the minds of some to say, if we can somehow on purpose give you like a little bit of this disease, can that make you immune to the bigger, much bigger disease, which in this most famous case was smallpox, the greatest killer of mankind among infectious diseases. So what does this vaccine have to trigger the specific B cells and T cells to fight this particular bad guy? It's got to have the antigens of that bad guy. How can it do that? Well, if you take the bad guy, this is how the early vaccines were made with that specific antigen, and you kill it or weaken it, maybe by heating it up, and then inject that into you, you hope that you killed it or weakened it enough to don't actually cause the disease with your vaccine. But now, with our ability to do genetic engineering and stuff, we can make all different kinds of vaccines where we only have the antigen in the vaccine. And so there's no chance that you can get the disease because you don't have the antigen attached to the bad guy that actually causes the disease. So anyway, I'll do this real fast because I signed up for this. This is COVID times when I'm making this video. I signed up for this vaccine trial. The vaccine that I signed up for is the Moderna one that has a piece of COVID RNA. And that gets injected into you, and there's a more fancy biochemistry to this, most of which I don't know. I'd have to look it up. But what your cells are convinced to do is to use this viral RNA to make a COVID antigen. I'll make it that shape, okay? So this RNA doesn't have complete COVID instructions. You wouldn't want to inject that into yourself. It would make complete COVID viruses. But it only makes the antigen, and therefore the first two, this is in phase 3, the first 2 phases showed great hope for this, and I'm not sure if I will be chosen for this, but I signed up for the phase 3 trial for that one. So antibodies again produce limited protection. Why limited? Because if you can't make more antibodies, and that's what the B cells do, right? If you can't make more, then again, those antibodies start to go away over time. They get eaten by your white blood cells, right? But if you get a certain disease, like rabies, for instance, rabies is a good example. If you get bitten by a rabid dog, one of the things you're given right away are antibodies against that rabies virus. Antibodies that were developed and grown in animals usually, to make this antibody thing, and that will give you some help right away. But those rabies viruses are also kicking your own immune system into gear. So you're making B cells and memory cells that can fight against the rabies virus. But rabies kills so fast that giving you some antibodies, providing you with some immunity that's called passive. Why is it called passive immunity? Because your immune system didn't respond to this thing and make these antibodies. You got them from an outside source. That's simply why it's called passive. How also can you get antibodies, passive immunity, besides being injected by ones that have been developed for the purpose? Well, this is something else that mom gives you. Some of these antibodies cross the placenta and go into your bloodstream. Also in breast milk, some antibodies go from mom to you. And so anything that mom has made antibodies against, you're going to get some protection from. But remember, you also have an immune system. But when you're first born, it hasn't kicked into gear yet, hardly at all. And so it helps. It has evolved a way for mom to get you a little bit of help to get you past those early vulnerable stages. So now, here's what we learned to do. If you take a cancer cell and you fuse it with an antibody producing plasma cell, literally fuse them together, you create a hybrid cell that's called a hybridoma. And because it was a cancer cell, it keeps multiplying over and over and over. This is in a petri dish in the lab. And because you merged it with a cell that makes a particular antibody, you now have a whole bunch of cells that make that particular antibody. And these kind of antibodies produced in this way are called monoclonal antibodies. I don't know why. Of all the basic stuff that they could have put in the syllabus for you to learn about the immune system, why they chose this is almost like a little trivial factoid, almost. Although it's a good fighter for diseases by making antibodies in large quantities. So what again we said was each one of you has some different molecules sticking out from your cell's surface. That includes your red blood cells, but also all your cells. And so that's why you can't get a skin graft, a kidney transplant, or a blood transplant that we would call a transfusion from somebody else, because your immune cells are going to recognize those somebody else's antigens as bad guys, and they're going to destroy them, and that immune response will be massive, and that'll be the septic shock kind. It'll also cause a lot of blood clots when those antibodies start sticking to that foreign tissue. So here we have a little red blood cell stuff. Now, we talked about this before, I think, in heredity, that we've learned the genetics of the ABO blood system. Remember, it was an example of codominance. If you have an A gene and a B gene, you make both kinds of proteins that here we're seeing now have sugars stuck to them. There's also carbohydrate sugars that are part of this ABO blood system. There's an A type, a B type, and again a type where your cells can have both or none. They chose to have you know this basic idea, that antigens, because these molecules sticking out would qualify under the definition of antigens, on the surface of red blood cells, stimulate antibody production in a person with a different blood group. So if you have type A blood and a type B blood gets put into you by transfusion, it's these molecules on the cells of that type B blood that your immune system, is going to identify as foreign. It's going to attack them, make antibodies, and when those antibodies start to attach to these blood cells, I tried to make a Y-shaped antibody there, sorry. When these Y-shaped antibodies start to attach to your red blood cells, blood clots are what kills you there. Okay, this is, I just said all this, so I'm going to skip this. You can pause on it and read it if you want.